All right, welcome back. Just a quick announcement that directly after this talk, we will be taking a group picture somewhere. We will let you know where. But then without further ado, let me introduce Weini Huang, who is coming to us from the School of Mathematical Sciences and Queen Mary University of London. And she'll be talking to us about mutation, accumulation in healthy tissues.
Sorry, could you please turn on the microphone?
Okay, great. So yeah, so if anyone in London, just let me know. We have a nice building with famous geometry tiles, and then we have a nice math biology seminar that I was organized, so you can give a talk in our institute. So what I will talk about is mutation accumulation in healthy tissue, but the model is a little bit more general, just what we applies to in terms of this ecological growth histories that you heard about this today from Allison's talk. So what I'm interested in is eco-evolution dynamics in biological system and applying to cancer. There are some personal background because I was doing master degree in ecology and I was doing really field work collecting elephant dumps. So I care a lot about the ecology of the systems. And I went to do a PhD in evolution theory. So I care about how does the ecological dynamics could potentially change the evolution future. So in old times, people always separated time scale between ecology and evolution because they think the systems always go to ecological equilibrium before the mutation arises. Then with more of the work we know about genetic mutations, people realize these scales can be in the same time scale. So they can impact on each other. For example, if you have interaction with species, this can change to the population size of the system. For example, typical prey-prey dynamics can get values of population size. In these values, you have different evolution future that you are in a population size that in the peak of these dynamics. So in the other way around, if you have different mutations in the system, different genotype phenotypes, can cause different equilibrium in the system. So they do have a feed loop between each other. So different system I work at, I have worked a lot in co-evolution system. In terms of cancer, I've worked a little bit recently about cancer and immune cells. If I get the time, I have one slide in the end, but if I have time, I will talk about that. But mostly I focus on looking at either a constant population or growing population representing healthy tissue or cancer. And we look at how different ecological effects will impact different genetic diversity. So which we mean about genetic diversity can be pure measuring allele frequencies or genotype or phenotype frequencies, or it can be copy number variations. You heard the talk of HitGap. So I worked quite a bit on the ecDNA, but in the human cells. So we look at the evolution of the copy number distributions. And also I'm working on the RNA copy number distributions in the system. But I'm not talking this, but I will give another talk that I might post online if you're interested. So then a lot of other work we worked with, which I feel more familiar, like more relate to the people here, is the point mutation accumulations. In terms of the distributions, is about the site frequency spectrum or single cell mutation burden distributions. So that's what I will talk about today. So I think this audience knows very well about that. We have already know that from David's talk that somatic mutations is important both in cancer and healthy tissues. And during the somatic mutations that is a stochastic process, you will collect some information that maybe you can use to infer evolution parameters that from the talks this morning you heard. There are a lot of theory work, for example, the famous work from Kessler and Levin in the PNS, they talk about the precise solution of the Lyra-Debrec model. And there are other people in the audience, there are Thibaut Anta's group, worked a lot about this clone size distribution of mutations. Yasmin also worked quite a bit on this topic, and also the talk of Kit and Jason. And there are a lot of people use this theory to apply to data. So the work of Kessler and Levin was applied by Trevor Rankin and Andrea Soto-Rivas group in foreign kind of selection strands in public cancer data. And they found quite a proportion of the tumor data can expand by neutral selection. And there are also the work from Sandy and Ryan do the same thing, but in the space. And of course, this or bulk sample where you would have things get entangled. For example, if you have birth rate and death rate, you hear from tomorrow's talk about speciation and extinction, those things are combined together. So if you get these phylogenetic trees or with the bulk data, sometimes it's very difficult to infer back discrete values between the birth rate and death rate. What you get is effective birth rate. But there are also work that you use multi-region sampling data. Benjamin Weini and Andreas Torriva by using samples from different space and tracing back and looking at how to distinguish the birth and death. So there are a lot of work that's related to this, but this is all related to tumor. And from the data point of view, that there's more and more people aware, we also need to understand not only happening in cancer, but what is happening in dynamics in healthy tissue as a control. So a simple example is that this in the skin data in this Martin Cariana 2015 paper, they recognize many oncogenes that you suppose only present in tumor, but is also present in normal tissue. So here is just example list about healthy tissues where people to look at either genomic data or even single cell data. So this first paper is the one, first and second is what Kit was talking about this morning as well. And also the Lee paper, well, they originally that they take bone marrows, they take 89 cells from the bone marrow of a single individual, they clone them, expand them, and sequence into clones. And then they look at the clonal mutations of these clones, which represent back on time, these 89 cells, they're private mutations which is only carried by single cells instead by the whole population. So they have, like the recent paper where we talk about, they have like 10 individuals, which more cells and basically doing the similar things. So there are other papers related to other healthy tissues, but not in the same, maybe like scale as these papers. So it's become more and more people aware, understanding the healthy tissue is important by understanding what is different between the healthy tissue and the tumor. So what is a little bit missing, I think, in the field is more theory work with how to look at the constant population. You heard the talk of Kate and other people, that's what I was focused on. So no matter that we are looking at healthy tissue or tumor, the common thing between them is that there are multiple statistical properties that are arising from the same stochastic process. So again, using a healthy tissue example, you will have a population that they are starting from a single cell, and this population experience a growing phase, and they reach to a homeostasis, and then maintain the homeostasis for a while. So this in these cells, so in this example here, in the end, for example, you have 3 cells, and if you look at the cells, you can look at different information. Here, every colored bar there is a mutation happening in cell division. If you look at these 3 cells there, you can look at how many mutations there and how many cells carry this mutation. So if every cell carry a mutation, for example, in this red block, then it reached the frequency as 1 if it's haploid, and it's 0.5 is diploid. And this is what we know about site frequency spectrum, or you call variant allele frequency in tumor, in cancer biology. But the same cells, you can also look at how many mutations you have in the cell. And this gives the single cell mutation burden distribution. You can also, instead of look at numbers, just look at how deep is the last cell compared with the first cell, how many divisions they experience. This gives the division distribution that Alison talked about, phylogenetic trees of the cells. So what we want to say is this, all this information is come from the same stochastic process. So what my PhD student Crystal did is that he introduced a dynamic matrix to integrate in these statistics and to see whether you can get the information from one statistics to the other. So let me go through the matrix for you. So this matrix YI, I means the time event. So like if a birth death happened, then I plot increased by 1. So in this matrix, it's dynamics because every row is a cell and every column is a mutation. So it's 1 if this mutation present in that cell, is 0 if the mutation is not there. So if you look at the information in every row, this gives you how many mutations that a cell carry. So which give you the blue columns that you have seen. That's the single mutation burden distribution. So if we know which mutations are rising at the same division, which is these gray areas, and then you just count how many gray areas a cell has, this gives you the division distribution, which is the phylogenetic tree information of cells. Now, if you look at column, which you sum over the ones, it tells you how many cells carry that given mutation. So this is the frequency of the mutation. So the orange line gives you the site frequency distribution. Any question related to this one? Okay, so I assume that everyone got it. So if you put this information here, you have three distributions. Orange is site frequency spectrum, and the division distribution, and the mutation burden distribution. And this distribution, if you think about data, they need different information. So this can be a bulk data, where you just have a bunch of cells and you only have the information overall, and you know the frequency of different mutations. But the later two cells, C and D, you did need single cell information. That's referring the two talks in the morning. So what we want to do is that since they come from the same matrix, you can just write down the matrix, because all the one need to sum up equal to the same number, no matter you sum across the lines or sum across the rows. So you see, for example, if we use here again, the BKI is the number of cells have K mutation at time I. So this is the single cell mutation burden distribution. And S again is the site frequency distribution, tells you how many mutations are present in how many cells. And you can see this, you need to write, if you write down some of the ones, they need to equal to each other. And this we can use to infer the mutation distributions from each other. So what we did is that we write down the recurrent relations of these different statistics change over time. So we write down the distribution of SJI plus 1, which means at I plus 1 time event, how many mutations are present in J cells. So J divided by the population size will be the frequency. And how this relates to the last time events of this distribution. And then what we want to do is that to assume different growth. For example, if you look at this macro chain there, so this macro then tells you the population size. So you start from, you can be 0 cells, 1, 2, 3, 4, and you have probability beta to have a birth event and the data to get a death event. So if beta is equal to 1 and the data is equal to 0, which means that you have pure birth. And because the I is the time event time, so because you have only pure birth, so at I plus 1, so at I time, then you get I plus 1 individual because you started from 1 individual. So by under the pure birth assumption that we can solve the expectation of the distribution for site frequency distribution, which has this polar shape, which is f to the -2. This result was also in the Kessler paper. Also, Yasmin has the same results, I think, in a different angle. And so it's well-known results. What we could extend is that if we can do approximation, use the recurrent relations to assume that if you have deaths, but your population is growing, so your death is much smaller than beta, and what is the distribution looks like. And we can do the same thing for division distribution. Again, look at the pure birth case where data is equal to 0, and we can get the solution, exact solution for the division distribution, like how many divisions a cell has in the I time event with the length L. And this results already known for a long time. Is the first paper we found is already in 1965, and a lot of people have repeated in their work and get the same results. And we can also do the approximation again in a growing population where birth is much larger than death. And what is interesting is that the single cell mutation burden distribution, which no one else was working before, with a good reason, because there's no data in this a while ago. So what we think about is that if you think about the division distribution and the single cell mutation burden distribution, they should be very related because they both need the information of the single cell. And what difference between these two distributions is that on top of the division distribution, the single cell mutation distribution should have another stochastics. Because what you had is that you had a cell, they can divide, there's some stochasticity related to birth death events. But per division, you have a random number. Normally people take from Poisson distribution. This is another stochastic come per the division. So you would think that to get the single cell mutation burden distribution, you need to consider two stochastics, one from the cell divisions, one from the stochastics per divisions because of the mutation number can vary. So that's what we thought as well. So we look at the situation, we write down again the recurrent relations for the BKI, which is the single cell mutation burden distribution. We try to solve it in a pure birth case. Well, we get exact solution. The solution was quite straightforward, that you have the product of the solution of the division distribution times the Poisson distribution. But if we look at the solution inside, we didn't see any variance appears. So we only see the mu, which the mean number of mutations per division, and the variance doesn't matter in the solution. So if we look at the growing population, well, data is much smaller than beta, we get a similar solution. So instead use the exact solution for pure birth of division, you just replace it by approximate solution in the first order to the solution of the single cell mutation burden distribution. And again, we don't have any variance in the solution. So this means that the final mutation burden distribution is mainly driven by the stochastics in the cell divisions, but not so much on the noise in how many mutations you get in the single per divisions. So you can have the same Poisson distribution that you have large variance or small variance, but they give you the final mutation distribution with the same distribution. So this surprised us a little bit because we consider both should play a role, but in the end, only one matters. So what we did is we want to test in our simulations whether we can recover the mutation burden distribution by just knowing the division distribution. So we have a stochastic simulation implemented by Gillespie algorithm. So everything is stochastic. We can record all the information over time. So we know the site frequency distribution. We know the division distribution. We also know the single cell mutation distribution. But we assume we don't know the single cell mutation burden distribution. And we only know the division distribution. Can we recover a distribution? So that's the procedure how we did it. So we have this, the upper parts here is a conceptual picture how we do it. So we do a stochastic simulation. We generate the division distribution. So how many cells experience how many number of divisions over time. And then in this division distribution, in each bar, so L dash means there are so many cells experience L dash divisions. And we take this information and use the Poisson distribution. So we use the mean of the Poisson distribution with this L dash to generate new distribution. And we do this for all the x-axis in the division distribution, and they will pull all this distribution together and generate a virtual mutation burden distribution. So which means that from here to here, we know the division history of the cells, we know the mean number of mutation per division, and we generate an artificial, like unknown distribution, which tells you how many mutations a single cell has. And we want to compare, is this distribution overlapping with the simulation results? So that's what you see. So here again, this is averaged from multiple simulations. Well, again, in this site, the purple one is the division distribution we have. And then if you look at here, the dashed purple one is the generated single cell mutation burden distribution coming from the division distribution. And the blue lines is our pure stochastic simulations. So you can see they fit very well together. So indeed, the variance of the mutation information per cell division really doesn't matter for the single cell mutation burden distribution, which we were not expected before we have our analytical results. So, and then we tested with other distributions, like supposing that in the simulations, you have uniform distribution per cell division. So the two daughter cells can, if you have a mean of 2 and between, of 10 between 1 to 20, you can equally get the same number of mutations in the two daughter cells, or you have a geometric distribution, but they all can give you the same conclusions. So no matter which distributions we use, you always can get the single cell mutation burden distribution just by knowing the mean and the division history. So I hope that I explained a little bit why we think these statistics that they really talk to each other. You can mathematically, analytically to see how you derive one distribution to the other. So the question is that, can we combine these statistical properties to get a better inference in real data? Is what people get used to use is that either you use the site frequency distribution or use other information to infer parameters. But if now we have both informations, can they improve the inference? So again, we use example of the growing population, but one details I missed is maybe here. So if we think about stem cell population, so the cells start from single cell and grow to a homeostasis, and you have cell turnovers afterwards. In the stem cell population, then you have this very important parameter is called symmetric division or non-symmetric division. So symmetric division means that you have a cell divided into 2 stem cells and stay in the population. And asymmetric division means that maybe you have a cell divided to another compartment, or if you have cell death. So if you only care about the stem cell population, then death and asymmetric division go to the later compartment is equivalent. So we have this system, that's what you see. So then your variant, so the site frequency spectrum might change over time, along in the growing history. So in the growing phase, you might have a different distribution compared with the homeostasis phase. And what we see is a sampling. So as examples, with the example that Kit talked about this morning, and I was also using the same data, is that they are sampling 89 cells out of you don't know how many stem cells there are. So the sampling is very sparse. So it's a very small proportion. So what we want to understand is how these different growing phases matters with the site frequency distribution, and what does sampling play a role on it? And then can we use some information from which statistics to infer which parameter? So we first look at our simulations here. So A is the simulation results. So from our work and also so many work before, we know there are boundary distributions. So again, this purple line here has the shape of f to the -2. So f is the frequency. It's a growing population. So if you have only pure birth case, the site frequency distribution has the shape of f to the -2. If you have a constant population, which means the cells stays in a homeostasis case, then you have f to the -1. So if we simulate the process of a cell growing to a homeostasis phase, we see a very interesting transit. First, the population will fast moving towards a homeostasis phase and stay very long there. So you kind of have a homogeneous transit between these two distributions. And then we look at the work in Sangha, where they have published some nice work in 2018. So they have the healthy esophagus tissue from 9 individuals. So they take different patches. You can think about every patch is a small independent population. And if we group this site frequency spectral information from this data, and you group them in different age, so like the gray area is the young people, and you go to more older people, and you can see this clear transit in the site frequency spectrum in the data as well. And then we were asking is what we are interested in is that in reality, those data are all sampled. So what does sampling play a role in it? So again, we look at our simulations. So the blue one is a full population where if you have all the information, and then the orange one is the sampled population. You see sampling shifting the mutation frequency upwards, which is quite understandable. So if you're sampling a few individuals from a large population, what you lose is the rare mutations. And we know that in a growing population, that's the highest number of mutations are all in a low frequency. So that's in this part, and they're lost in the sampling. So that's the impact of sampling. But what you can do in theory is what we observe actually is this distribution. And we want to understand how these two distributions are related. So what you can write down is that you can simply see how you can inferring the true distribution of the whole population based on the observed distribution if you could know the population size of the true population. Obviously, you will know the sampling size. So you know you take 89 cells, so the sample size 89. But what is missing is population size. So this is a really interesting property that maybe you can use the correlation between the sample distribution and the true distribution to referring back. But before we go that, I was saying, can we integrate in different statistics? So the last slides, I was talking about the site frequency spectrum, but I was talking a lot about the single cell mutation burden distribution that we worked theoretically a lot. So we can do the same thing and look at how sampling matters for the site frequency spectrum. So actually it doesn't matter so much. So sampling has a strong impact in the site frequency spectrum, but not so much in the single cell mutation burden distribution. So you can see again here, the gray area is the average of the whole population. So suppose you know the population, you pull all the information of every cell, so look at how many mutations each cell carry. This is the gray area. And then the blue one is a single realization of the whole population. And then the orange one is the sampled population. So we use the same parameter as the experimental data. They have 89 cells, and we simulate the population to 10 to the 4, and we sample 89 cells out of 10 to the 4, and generating this orange distribution we have. You can see these two distributions, they're overlapping very well if you really do a random sampling. So which means that single cell mutation burden distribution seems not so much impacted by sampling. So this helps you to infer some parameters. So if we use the single cell mutation burden distribution to infer the mutation rate, so how many mutations you get by division, and you can see using the whole population information, which is the blue distribution, or using just the spare sampling individuals, which give you the orange distribution, the inferred parameter distribution very well overlapping with each other. So this confirms the other distribution here. We can use, maybe if you have quite a sampling effect, using the single cell mutation burden distribution can give you much accurate information of mutation rate than compared using the site frequency spectrum, which you don't know the population size. But what can't single cell mutation burden distribution do alone is that you can't distinguish the asymmetric division and symmetric division rates. These two things are entangled together with the mutation rates. So again here, so this now we are looking at how does the single cell mutation burden distribution look like in the data. So this is the work by Peter Kamper's lab. So that in 2018, well, they have these 89 cells gathered from one individual from the bone marrow. So what they see is that in that data, on average, every cell has around 1,000 mutations. And these mutations have roughly a compound Poisson shape of the distribution. So we use a compound Poisson distribution to fit the data at the time is because we haven't got an analytical solution that I talked about before. But these two solutions are very similar. So it's our kind of educated guess. So what you can do is that this is the single cell mutation burden distribution, but we also know if we pull the information in the single cell, it is a small population. So you have a sampled population with 89 cells, and you can look at the mutation frequency in these 89 cells. So you can group to the site frequency distribution of these 89 cells. And because if you know N is the population size, S is the sampling size, which is 89 in this case. If you know N and S, you can write down the relationship of the sampled distribution, which is what you observe, with the true site frequency distribution, which is Vj. And the thing is that, okay, now we know this distribution, which we observed, and now we know the S, and now we know the single cell mutation burden distribution to inferring the mu. Can we narrow down the guess of N, which if you only have information of one, you'll get a huge larger range. So what we did is that we used the single cell mutation burden distribution, infer the mutation rates and division rates that coupled. And then we fit the corrected observed WAF, which is the sampled WAF, and generated by assuming different population size N and the asymmetric differentiation fractions. And we see what is the best combination of N and asymmetric division fractions give you the most observed variant allele frequency distribution. So that's what we see. So with the same data, we can see we vary the population size that we don't know as n, and we vary the symmetric division rates between 0 and 1. And we can see that you get a best fit curve. And what we like most is because in this curve, give you a natural maximum value of n that you can have to generate this data. So which means, what is the maximum value of n? This is the maximum value of the stem cell population size that you can have to have such observed data. And this value in this data here is 2 times 10 to the 5, which is very agree with what everyone was talking about, but no one has a real number with it. So we kind of think, okay, you can the single cell mutation burden distribution, you can infer the mutation rates and the correlation of mutation rate with symmetric division rates. And then you use the sample, the variant allele frequency distribution to get the population size. So do we have, yeah, have a bit time. So I talk about a little bit side project of our work. So when we're doing this work, we recognize when we do our simulations, even in a constant population, even we introduce a mutation rate, which means that mutations constantly arising during the process, but mutations doesn't get fixation or doesn't reach to a high frequency at the same time. So the arising time for them is continuous, but the time they reach to a certain frequencies is not randomly distributed. So what we observe is that we call as mutation waves. So what is mutation waves? Mutation waves, what we define is when multiple mutations reach a given frequency at the same time. And you can think about this figure here. This is time, and this is the number of mutations in the population. And what you can see is that those fixed mutations, they will go with jumps. So this means that you reach the mutation frequency as 1. And this is already actually worked a lot historically by other people. But what was haven't been worked about what happens before fixation. So is obviously if those mutations reach to fixations at the same time, multiple, they must reach to certain frequency at the same time as well. And how this impacts our understanding of data and all these variant allele frequency distributions. So what you can see is here, if you look at time, and you look at the number of mutations at a given frequency here, let's say 0.75, and you can see this, there are a lot of empty spots here, which means that in a single simulation, you have many times there's just no mutations reach to the frequency of 0.75. And if you think about what variant allele frequency, what I was talking about is that if you look at a single time here, so suppose 2,500, and if you look at all these distributions at different frequencies and you put them together, that's the variant allele frequency distribution. So this means that if you have a lot of time that you have no mutations given in a given frequency, that means that your variant allele frequency distribution in signal realizations is not a continuous distribution. They have some bimodal case. And what we calculate is expectation. So either you have a kind of events happening long time, you average over time, or you have multiple repeats of the same evolution trajectory and average cross trajectories. But what matters is that what is the single cell trajectory differs from the expectation. Because what we have in every data is a single trajectory, and you look at the data in a given point. You're not looking at data average over time. You're not looking at the data average from multiple samples because they are different individuals. So that's why we look at for different frequencies. So this is the mutation frequencies. If you look at the simulations, how many percentage of time those single trajectories doesn't have a mutation in a given frequency? Very obviously, if that's the frequency is higher, there's more time, but there's just no mutations have frequency in the over time. The reason is that if you think about mutations have frequency of 0.75, if there are two mutations, they must overlap in some way in the same cells, because you can't have one cell clones carry this mutation and again another cell clones carry the other mutation. They all reach the frequency of 0.75. So they must overlapping in some point. So those mutations shared in the same cell, And because cells is the division unit, they need to spread in the population. This causes this kind of discrete distributions in variant allele frequencies. So if we, instead of look at the variant allele frequencies, we can look at the wave frequency distribution. Basically, you just take the time, take away the information while the distribution just have no mutations at a given frequency. You can see they overlap very well in low frequencies, but deviate a lot in high frequencies. So what this means that, of course, when we look at data, we look at the variant allele frequency, we compare with our theoretical expectation on the average. And when we see a mutation or area, a much higher frequency, so some mutations much higher frequency than expectation, quite often you will draw the conclusion that is caused by selection, because selection make these mutations in a much higher frequency than average expected in a neutral evolution case. But because this discontinuity of in the single realization different from the expected side frequency spectrum, this can be another source why in the single simulation, single trajectory data that you have observed high frequencies than the expected value, especially in the high frequency case. So I think maybe I have, 3 minutes. So I come to the point that I also talk about more than just the cancer cells, because we have worked so much work about mutation distributions. And I have worked a lot of work about co-evolutions. So I will sort of bring these things together to see whether we can use the mutation distributions to distinguish the interactions between the cancer and the immune interactions. So what we had is a very simple gene for gene model. So there are a lot of work in the past 1 or 2 years about modeling the new antigenetic mutations in the cancer cells. But what people haven't done is that the immune reactions is always considered as a average value in the population. So, but in biology, when people talk about host parasites and other co-evolutions, people quite often use gene for gene model, where you have the antigenetic mutation in the cancer cells need to be intact with certain specific effector cells that can recognize these antigenetic mutations. So we introduced this gene for gene model, and well, we also have the mutation accumulation process, and we look at if the interactions, so the interaction parameters between the immune and the cancer cells are strong or weak. Do you see some signal difference in the molecular level where you look at the mutations of the population? So we didn't observe a very strong deviations in the side frequency spectrum, but we do see some deviations in the single cell mutation level information, which means maybe the single cell data could be used to inferring how strong the immune cells are interacting with the cancer cells. So comes to here, I give my summary. So we have a framework with the dynamic matrix to unite variant allele frequency distribution, division distribution, and the single cell mutation burden distribution. And then we talked about how sampling will shift the variant allele frequency distribution. And then we talk about how can we use different statistics and use the bulk and the single cell data to help narrow down parameter inference, especially in our case is the population, true population size of the stem cell population. And we talk about the stochastics in the single cell trajectories might have different discrete distribution compared to the continuous distribution on the average, and then how this impacts to understanding the data. And then I talk about the mutation burden information may infer cancer immune interactions. So up to here, I would thank my collaborators and thank you for listening.